Welcome back. Today we're actually going to discuss basics of cell biology. We're going to focus first on the cell theory and then we're going to move into all of the cell parts. First, I want to bring you a little bit back into history. In the 1600s, van Leeuwenhoek improved the simple microscope by making just a new lens that was a higher magnification and allowed people to observe a lot more detail. I always repeat in class that we're always limited by technology, and this is a prime example of why. So I just want to run through a nice little breakdown before we get into a little bit more history. So today we're going to really talk about cells, and there are two major types of cells, the prokaryotic cell, which is unicellular and does not have any membrane-bound organelles. And a good example would be bacteria, and going even further, a specific type of bacteria would be E. coli or anthrax. Versus the other side, we have eukaryotic cells, which can be unicellular or multicellular, just like our reading said. They have membrane-bound organelles, and we have two major examples, plant and animal. For plants, you can think of a tree or an onion, and for animals, you can think of the human, tiger, dog, or even protists, which are single-celled organisms. Before microscopes, people had no idea tiny little microorganisms existed. And they used to often think that they were curses that people um, gave to other people, or they were supernatural spirits trying to get back and get people in trouble. But in actuality, it was just a microorganism causing the person to get sick. Moving forward, in the early stages of the cell theory, we had this scientist named Robert Hooke. He was also from the 1600s. He used a compound light microscope to actually study a cork, the cork that you put in a bottle or a wine bottle or whatnot, and it's actually made from plant cells. So he saw underneath the microscope that all of the cells looked like little boxes. Now, if you're wondering where he got the name for these tiny little cells that he came up with that reminded him of boxes, he remembered that they reminded him of the rooms that monks that lived in monasteries stayed in. Very simplistic, little tiny little 4 by 4 square, very basic. That's what he reminded him. That's why oftentimes you think of plant cell being very structured. So Hooke observed that cells are the basic units of all living things. Without a single cell, you can't be considered living. So Robert Hooke wasn't the only scientist to help out this field of science. You have Robert Brown from the 1800s that found the cell nucleus. He finally identified it. Matthias Schleiden, 1830s, German. He observed all plants are made of cells. Theodor Schwann, another German, that all animals are made of cells. And then you have Rudolf Virchow, all cells are from other cells. So the whole entire idea of the cell theory was kind of a progression of many scientists. So the first part of the cell theory is that all organisms are made of one or more cells. So on the right-hand side, you have tiny little bacteria, they're single-celled organisms, versus the left side, you have a big plant that actually has a beautiful flower over here, and that's made of multiple cells. Second stage is the cell is the basic unit of function. Even if the organism is big or it's simple, its simplest component can carry out any metabolic activities in the cell. Now, metabolic activities include any chemical reaction in the body or in that cell that is going through that process in order to maintain homeostasis. Lastly, because there's only 3 stages in the cell theory, all cells come from pre-existing cells. To get another cell, you must divide from an existing cell. And you're probably wondering, hey, some of my previous science teachers mentioned that there were some things that really didn't fit in there. So, we're going to talk about viruses. Viruses are not made of cells, but they do have genetic information. Some viruses have DNA, which is double-stranded, and other viruses have RNA, which is single-stranded. And they can reproduce, but they need a cell's help. They need to hijack a living cell and use their organelles to produce more viruses. The other problem with this theory is that mitochondria and chloroplasts are organelles that have their own genetic information and can reproduce on their own. So you're at a crossroads here and you don't really understand why, but we're going to actually explore some of that later on. And then The simple question of where did the first cell come from? Almost like did the chicken or did the egg come first? Who knows? There are some theories. We will discuss them in class at a later date. But generally, I'm going to go into cellular organization next. So if you were going from smallest to largest, you have to think of all the little components that go inside of a cell. These are the cell organelles, and then a bunch of cell organelles together would create a cell. A bunch of cells together, you'd end up with a specific type of tissue. A bunch of tissues together, you would get an organ, and a bunch of organs together, you get an organ system, and then a bunch of organ systems together like us, you're going to get a multicellular organism, which is your largest you can go. Classifying basic cell types. Okay, in our reading, we learned about two specific types, and we're going to explore them just a little bit now. So cells have structures inside called organelles, or tiny organs, is a good way to remember it. In some cells, they are surrounded by a membrane, but in prokaryotic cells, they are not membrane bound. A great example of this is bacteria. They have DNA just floating around inside them. There's no membrane that binds it into a specific organelle. Then we come to the types of cells that we are made of. We're eukaryotic cells. We can be multicellular, we can be single celled, it does not matter, but the distinct, interesting thing is the fact that we have organelles that are membrane bound, okay? And they act as compartments with specialized functions. So each of these cell organelles have a specific function in order to maintain homeostasis. So comparing eukaryotic to prokaryotic cells are 1 to 100 times bigger than prokaryotic cells. We have membrane-bound organelles with distinct jobs. Not all eukaryotic cells contain a cell wall. Only plant cells have cell walls, and some single-celled organisms may have cell walls as well. Prokaryotic cells are much, much smaller than eukaryotic cells. Their DNA floats in the cytoplasm. In addition, inside the cytoplasm, metabolism happens, and also cell wall always. Believe it or not, antibiotics destroy the cell wall of bacteria. That's how they generally work. Little fun fact. So two different types. What are these two different types? We have plant cells on the bottom left, and we have animal cells on the top right. So animal cells, they have no cell wall, smaller vacuole, no chloroplast, and they have centrioles for cell division, and we'll learn more about centrioles when we get to mitosis later on in the year. Plant cells, on the other hand, they have rigid cell walls, huge vacuoles, and you should think about a juicy salad. Every time you eat salad and you chew and chew and chew, it's getting more watery. You're actually bursting all of those vacuoles with your teeth. They have chloroplasts that contain a pigment called chlorophyll for the site of photosynthesis. Now, your most important organelle is your nucleus. This is your control center of the cell. It's important because it contains all of the information that is needed, all the DNA that is needed to make proteins. Okay? It's kind of like the blueprint to make that protein that you need to do other things. In addition, the instructions are containing chromatin, which are just loose strands of DNA. You can kind of think of this as like a piece of cooked spaghetti kind of floating around. But then when the loose spaghetti or the chromatin actually coil up and get tightened, that's where we get a chromosome from. So when that chromatin condenses, that's how you get a chromosome. The nucleolus is the organelle inside the nucleus that makes ribosomes. Ribosomes make proteins based on the DNA information. They actually read a code, and we're going to go through this whole process much later in the year. The nucleolus makes ribosomes, which makes proteins. That's a general statement you have to remember. Cytoplasm is a clear gelatinous fluid in the cell. By gelatinous, I mean almost like jello. It's a jello consistency. It contains a lot of water, okay? Now, animal cells and plant cells, we both need water, so you're going to see that it's very fluid. The endoplasmic reticulum, also known as the ER, is a series of highly folded membranes suspended or just hanging out in the cytoplasm. It looks like an accordion, and if you don't know what that is, it's an old school instrument with a lot of folds in the center. It adds a lot of surface area. Okay, surface area is important because the more surface area you have, the more chemical reactions that could go on at the same exact time. And if you ever wonder why surface area is important, think of taking a cube of sugar and melting it versus taking powdered sugar. But the powdered sugar has a lot more surface area than the cube of sugar, and it's going to dissolve a lot quicker in water. There are two different types of endoplasmic reticulums. You have the rough ER, which actually contains some embedded ribosomes that are attached to the surface, and this is additionally where protein synthesis is going to occur. And then in addition, you have the smooth ER, which there are no ribosomes, but its main job is to produce and store lipids. And in our next unit, biochemistry, we're going to learn a little bit more about lipids. Ribosomes, site of protein synthesis, one of the most important questions the exam may ask you over and over and over. It puts together different amino acids into polypeptides or proteins based on RNA instruction. I know that sounds like a lot of science words. We're going to get over it. Amino acids are just the building blocks of proteins. When you eat meat, your body's breaking all that meat down into amino acids, and then your body is taking those, each cell is taking those amino acids and putting them together in a new code or a new arrangement to give you the protein or enzyme that you need. That's what we call the polypeptide chain. And these are ribosomes can be found on the rough ER or floating around in the cytoplasm doing their job. The Golgi apparatus actually looks like a stack of pancakes. I know I'm going to be making a lot of food references. You're just going to have to deal with it for the rest of the year. The stack of tubular membranes actually sort proteins into packages and puts them in a membrane-bound vesicle that can be sent to another cell or just another part of the individual cell. Serves as like the shipping warehouse of the cell. Vacuoles, they're membrane-bound sacs. They store water, food, and waste. Much bigger in a plant cell because there needs to be a lot more storage than they are in an animal cell. And they serve as the storage room or the pantry room in your kitchen. Lysosomes, organelle containing digestive enzymes. It's like the trash compactor of a cell. It digests old organelles, food, engulfs viruses and bacteria, or cellular suicide. Some cells actually do, when they know they're about to die or they're not functioning properly, they will go to a lysosome and they will get eaten alive. They are membrane bound, so that way the enzymes don't digest the actual cell itself. Lysosomes can fuse with a vacuole and dump the digestive enzymes inside just to break down all the food and waste so they can be removed from the cell. Chloroplasts are dish-shaped. They actually capture inorganic light, sunlight, and make organic chemical energy in the form of sugar. This is what plants do through the process of photosynthesis. Has folded inner membranes for more surface area to trap more sunlight. In addition, stores green pigment called chlorophyll, and this is only found in plant cells. Mitochondria, very, very important. Powerhouse of the cell, converts organic energy molecules such as glucose from photosynthesis or ingested food to usable energy called ATP. Mitochondria contain an outer membrane, but the inner membrane is highly folded, and that adds a lot of surface area for more chemical reactions. This is actually the site of cellular respiration. Now, the theories of endosymbiosis is where mitochondria and chloroplasts have their own distinct DNA. So they believe that mitochondria and chloroplasts were their own living organisms that somehow found it beneficial to be working with other existing cells, so therefore they became one in their own. Centrioles are microtubule organelles. They are found in pairs, used in cell division only in animal cells. Then I'm going to bring you to cilia and flagella, made of microtubules as well, used for locomotion or movement. Helps unicellular organisms move. So cilia are short hair-like structures that move like a rowboat, and a flagella is more like a tail, come in one or two tails at the time, and they kind of whip back and forth. Your cell wall is found in plant cells, fungi, bacteria, inflexible, protects cells, structure, and support. And in plants, it's made of celluloses, which is a type of carbohydrate. In addition, it has pores, which lets molecules enter the cell, but it is not selectively permeable. Lastly, you have your cell membrane, which it surrounds all cells. Even if there is a cell wall, there's still a cell membrane. Keeps the cell in balance and maintains homeostasis along with everything else. Controls molecules that get in and out of the cell. Necessary materials in, waste and excess materials out. So does not always prevent harmful things from entering, such as viruses or toxins, which can actually harm and disturb homeostasis in the end. I hope this lecture helped. Good luck.